recording this computer. Okay, so good afternoon, ladies and gentlemen, and thank you for tuning in to another Risk Institute online session. Today, we're especially fortunate to have with us Professor Stephan Munch, an adjunct professor at the University of California, who will talk to us about his research on nonlinear dynamics for ecosystem forecasting and management. So the presentation will be around 45 minutes, around 20 to 30 minutes for Q&A. And please, could you make sure that your cameras are muted, sorry, your microphones are muted even, and leave any questions in the chat, or virtually raise your hand once the session's over, and I'll select them on behalf of Professor Munch. So Professor Stephan Munch is an adjunct professor at the University of California in the departments of Applied Mathematics and Ecology, Evolution and Behavior. And his research interests include nonlinear dynamics, Bayesian data analysis, and ecosystems management. So without further ado, if you'd like to share your slides and tell us a little bit about yourself, that'd be great.
All right. So let's see, how do we do that? Share screen, share. Can you see it?
Yeah, we can see it.
All right. First, let me say hi to everybody, and thank you to Scott and Francis for inviting me and patiently putting up with several delays before actually getting this on the calendar. And thanks to all of you for tuning in. I understand that you could be at a pub right now, and so I appreciate you coming to listen. So I'm going to talk today about, my slide does not seem to be advancing. There we go. Okay, got it. So I'm going to talk today about quantitative approaches to forecasting in ecological systems, the ultimate goal being ecosystem management. And to give you a sense of some of the applications, fisheries, which is my main gig, is a $200 billion a year industry that supports nearly 2 million jobs and provides 1/3 of the world with its primary source of protein. And the big questions there are how to set sustainable harvest limits when there are many species that are being harvested simultaneously. We also do a little work in agriculture and forestry, where pest outbreaks result in substantial damage every year. And between losses and control measures, pests cost roughly $70 billion a year. And the big question here is whether we can anticipate pest outbreaks and find cost-effective control policies. Ecological extreme events such as harmful algal blooms, fish kills, and forest fires are another area where improvements in prediction are important. Now, when we think about forecasting multiple interacting species in a quantitative way, we often start by constructing ecosystem models, and the practice of doing this usually involves the first step being assembling a team of experts who can help make the list of species and identify who eats who and other interactions. But having been involved in this sort of thing, it's clear that the list you end up with depends a lot on which experts you've invited. And the more people you invite, the more complicated the diagram ends up. And ultimately, that's because real food webs are really complicated. Ecosystems are composed of tens to thousands of species. And even if we had counts on all of them, there would still be lots of things left out, such as non-consumptive interactions, trait variation within species, such as age, body size, etc., genes, epigenetics, and lots of other stuff. And so when we think about modeling ecosystems, we could think about dividing the world into two classes of state variables, those we observe and those we don't. Now, critically, unlike other sciences, there is no fundamental theory or first principles to help us rigorously determine what things can be left out or what the consequences of leaving things out is. And so very often, although we know real life is more complicated, we usually often in ecology just end up writing down models strictly in terms of the observables and treating everything else as noise. And my argument today is going to be that maybe this isn't the best that we can do. So to be clear, we need to account for both the unobserved state variables and the fact that most of the time we don't know enough to write down a model for the system. So to deal with this, the approach that we've taken for the past few years is based on Takens' theorem of time delay embedding. Now, Takens' theorem says that if we have an m-dimensional dynamical system that's governed by a smooth map, which converges to an attractor A of dimension D, which is less than m, Then the collection of e-dimensional lag coordinate vectors of a generic observable from that system is an embedding of A, provided that E is greater than 2D. At this point, you're probably wishing you went to the pub instead, right? Well, honestly, this wasn't supposed to be illuminating. I just put it in here as a joke. So the practical upshot of Takens' theorem is that we can compensate for unobserved state variables using lags of the observed state variables. And here's an illustration of this. The 3 panels in the center show a time series that's being generated by a 3-species food web composed of a producer, a grazer, and a predator. Now, although the time series themselves look complex, when we plot the contemporaneous points in the XYZ phase space, that's the thing being traced out in black, we can see that they trace out a fairly simple shape. That's the attractor for the system. Now, alongside that, we have another set of axes and a red shape being traced out. And those axes are composed of lags of the producer now, the producer a step into the future, and the producer 3 steps into the future. Now, clearly there's a one-to-one correspondence between the shapes that were traced out on those 2 different sets of axes. Now that's cool and all, but what could we do with this? Well, if we had the attractor in hand and we knew, say, where we are now, we could find some nearby points on the attractor, look at where they went into the future, and use that as our prediction. Now, if we repeated that exercise from lots of different initial starting points, we're effectively constructing a discrete time model for the system. Obviously, we can play the same sort of game with the attractor in delay coordinates. And if we did that, we'd end up with a model for the next state solely in terms of lags of the producer or lags of the thing that we observe. And so this approach tackles both the problem of missing state variables and the fact that we don't necessarily know what equations to write down. So Takens provides some justification for writing models in delay coordinates, but it doesn't tell us what to write down. And this we need to approximate from the observed data using some sort of data-driven scheme. This could be either a nearest neighbor approximation, a local linear model, basis expansion, neural networks, if you're especially cool, or Gaussian processes. And most of the results that I'm going to show today are based on Gaussian process regression. So here's a little background on Gaussian processes for function approximation. The Gaussian process is a continuous generalization of the multivariate normal distribution. So it's characterized by its mean and covariance functions. So we typically set the prior mean function to 0 since we don't know much ahead of time about the shape of the delay embedding map. The covariance function, on the other hand, is where all the action is, and that controls the wiggliness of the possible functions. And that's illustrated here on the right. So for those who are interested, we generally use a tensor product of squared exponential covariances with distinct length scale parameters for each input. That's these phi i's shown here. And some sample paths are shown here. And the main effect of the length scale parameter is to control the wiggliness of the possible functions. And so I'll tell you a little bit more about that in a minute. So the idea here is that once we've set up our Gaussian process prior, we will update it with data. Since in the prior, our mean function is 0, the mean function is constant. It's a flat line here, and the uncertainty bands are wide. But as soon as we have some data and we update, the line moves in the direction of the data and the uncertainty band bunches up. And as we add more data and we condition on more data, we get something that starts to look like a function. So there's one more nice thing about this Gaussian process setup, which is that when the length scale parameter for a given input goes to 0, that input effectively drops out of the model. And so we can make use of this by setting a prior with a mode at 0, when that's shown here. And that leads to automatic relevance determination, which is illustrated on the right here. So the surface here is the fit without automatic relevance determination to this set of white points. And as we impose the automatic relevance determination, we find that one of the axes drops out, and that's right in this example. And one last thing, although my Bayesian colleagues freak out every time I say this, we generally make our predictions by fixing the hyperparameters at the posterior mode. And although I recognize that this underestimates the uncertainty, it's usually a reasonable approximation, and it saves us a ridiculous amount of time doing MCMC. And so with all that set up behind us, Here's an application to some fisheries data. So we looked at time series from 185 different fish populations, representing 70 different species from 26 families. Each of the time series that we looked at had at least 20 observations. And for comparison, we fit the standard one-dimensional parametric model that's widely used in fisheries management. And there are 3 or 4 of those that we tried. Okay, so now rather than show individual fits, I'm going to show you just a summary of all 185. The axes here are the scaled mean square error. So that's the prediction variance in leave one out forecasts divided by the total variance. And so the horizontal axis is the scaled mean square error for the Gaussian process embedding model, and the vertical axis is the error for our traditional 1D standard fisheries model. So the scaled mean square error, a value of 1 is what we'd get if we just used the mean for making predictions. And so, anything bigger than 1 means the model stinks. Okay, so and the diagonal line is the 1-to-1 line. Okay, so here are the points. From this, we can see that most of the time, the standard model that's currently used in fisheries has more error than the delay embedding model. In fact, the Gaussian process is better in 90% of the series that we looked at. On average, the prediction error is 25% less. So looking at this another way, using a 1D model to make our inference inflates the apparent noise. And this has obvious implications for any probabilistic projections we might make, such as evaluating the probability that a fish stock will recover from being overfished, or evaluating the probability that it will go extinct in the future. Now, we've applied this sort of thing, this delay embedding stuff, in lots of other cases. Here are a few highlights. Through a lot of simulation work, we've shown that the embedding approach is usually just as good as fitting a correctly specified parametric model. And it's far better than fitting the wrong model. We could use the fitted Gaussian process to construct near optimal control policies. And partial derivatives of our fitted models can be used to measure context-specific context-specific interaction strengths. But every time I talk about successful applications of this stuff, some cheeky bastard points out that delay embedding assumes that the system is stationary, and what about climate change? And so rather than get to the end and have climate change be a big question, let's just sort of address this head on. And so we've all seen evidence of climate change in the form of extreme weather events, droughts, fires, and we've probably all felt sorry for this polar bear in the upper left. So despite what half of my country seems to believe, climate change is real, and we need to acknowledge that. But the typical conceit is that mechanistic models will do better at extrapolating as the climate changes compared with the phenomenological delay embedding models, which are often dismissed as black box models. And so let's think about this a little more carefully and take a look at what climate change might mean for delay embedding. So here's that same 3 species model that I used to introduce the delay embedding a few slides ago. Now let's imagine that all the species in that model have vital rates which depend on temperature, and then we'll let the temperature change. So honestly, I don't know what I was thinking when I made the temperature go down, but it doesn't matter. It should go up, but either way, this monotonic trend in temperature has the effect of making the attractor fuzzy. Now, obviously, there's still enough shape there that we could potentially use this to make predictions, but they'd clearly be less precise. That is, if we identified a ball of points, there are lots more, there's a much bigger range of possible next states that are feasible with the information that we have. So, in fact, to make this a little bit more concrete, we could evaluate the decay in prediction accuracy as I change the rate of warming in this model. And at least for this model, we can see that the prediction accuracy decays roughly linearly in the rate of environmental change. So yeah, climate change is likely to be a problem for these delay embedding methods. So what can we do about it? So when I started out the talk, I said that we don't have a lot of fundamental theory to fall back on in ecology, but one thing we do know an awful lot about is the effects of temperature. The metabolic theory of ecology is probably one of the most successful theoretical syntheses in biology. And the basic argument is that metabolism and other vital rates scale as the 3/4 power of mass and have Arrhenius temperature dependence, where the activation energy is typically about 0.65 electron volts. So Jim Brown, Jeff West, and colleagues have done a great job of showing that this applies not just to metabolism, but also to somatic growth, fecundity, mortality, population growth, etc. Now, most of the support for this work comes from comparisons across species, but what we need in this time delay embedding framework is something that applies to changes within species. And so here's an example of a study that we did looking at how well the metabolic theory explains variation within species. We went and compiled estimates on lifespan from multiple populations that are living at different temperatures, different average temperatures. And that allowed us to estimate species-specific activation energies. And that's what's shown here. Each dot is an estimate for a different species, ranging from frogs to bivalves. And although there are clearly some exceptions, most species come reasonably close to the expectation of the metabolic theory. And that's the black line shown in the middle. So to a first approximation, we could argue that lifespan and the time scale for life more generally scales predictably with temperature. So the next question then is how can we build this very specific temperature dependence into our equation-free, non-parametric time delay embedding model? So let's return to our generic multi-species model, which is now driven by temperature. The fundamental argument of the metabolic theory of ecology is that the temperature dependence is separable from the remaining state variables and that it's highly similar across species. So this implies that we could introduce a physiological time given by the cumulative temperature dependence, and that by changing variables from calendar time to physiological time, we'd end up again with an autonomous system. And so we're back on firm footing from the point of view of delay embedding. We'd integrate this to obtain a model for dynamics on a discrete physiological time step, and then turn that into a model with metabolic delay embedding, where the inputs are equally spaced in physiological time rather than in calendar time. And what this means intuitively is that we'd want to take bigger steps. We want to have the lags farther apart when it's cold and things are moving slowly, and the lags far closer together when it's hot and things are changing fast. To see how this plays out, here's our 3-species simulation forced by temperature, but now the temperature has some noisy seasonality as well as a trend. The attractor is shown here using traditional fixed time step coordinates. And it's a bit messy as expected, but replotting this using a physiological time step cleans things up nicely. And so that's a simulation. Of course, we want to know how this applies to some real data. So these are the blue dots are time series abundance of the spiny water flea. That's this scary looking thing up in the top right. These are sampled weekly from Lake Geneva. The top panel is the temperature time series over that period, which has some warm periods, some cool periods. It's obviously a little bit noisy. And so here is a comparison of the prediction accuracy with the black line showing the prediction accuracy using standard fixed flag embedding, and the red line is what we get using a metabolic time step. And just to be clear, the prediction accuracy here for each point is made by leaving a year out in either direction. And so we're not just smoothing the time series. This is reasonably close to pure prediction. And so here's what the predicted time series looks like. It's not bad. I mean, it's kind of jagged, but overall it's pretty close. It gets the shape reasonably closely. And maybe more importantly, it does a good job at capturing this sort of off-season high winter abundance in 1991, which is sort of a surprise. So, so if it was really just mirroring that sort of average cycle, that wouldn't have happened. Of course, this is just one species, and so we really want to know whether this generalizes. So So here's a quick summary of an application across 19 time series. These include time series of phytoplankton, zooplankton, and terrestrial insects. And the bottleneck here is that we really need data to be sampled at high frequencies in order to be able to interpolate to a metabolic time step. And so these are all the time series I was able to find that are sampled on weekly to monthly time step. So I've plotted here the percent improvement in prediction accuracy on the vertical axis, and that's so the percent improvement relative to using a fixed time step. And the horizontal axis is seasonality, measured as the coefficient of variation in temperature. In 13 of the 19 populations that I looked at, the metabolic time step improved prediction accuracy, with the biggest improvements happening for species in highly seasonal environments. So I think this is pretty encouraging. But as you all know, trends in temperature are not the only issue with climate change. And in particular, this separable dynamics assumption can't, and the metabolic theory of ecology, can't account for climate-driven regime shifts, such as the one illustrated here. So in panel A, we have a kelp forest, and on the bottom in panel B, we have the same location a little bit later in time covered by turf seaweed. And the time series on the right shows the relative cover by kelp versus seaweed. And as the authors points out, there's a rapid switch in cover coincident with several years of high temperature, those the red bars sticking up. And they point out that this probably temperature-driven switch in temperature-driven change in algal cover. Now, this sort of regime shift is not something that we're going to be able to get with the metabolic theory argument, but it is something that's had been a hot topic in ecology for about a decade. And a lot of effort has gone into deriving leading indicators of regime shifts. Now, these are all based on codimension 1 bifurcations, that is one parameter changing. And they generally let us know that a new regime is coming, but not what a new regime will look like. On the other hand, there is a lot of retrospective work characterizing different regimes. And a very nice recent example of this is one that points out that distinct regimes have, not surprisingly, different attractors. And so one question we might ask is, if we really are looking at a codimension 1 or a bifurcation or a 1 parameter change, how many regimes do we actually need to see before we can predict what the next one will look like? So here's an example of what I'm talking about using the logistic map. So this has been a staple of theoretical ecology since at least the 1970s. The bifurcation diagram is shown on the right. As we increase the population growth rate, we go through a sequence of period doubling bifurcations into chaos. And then to see whether we can use our Gaussian process embedding to predict regime shifts, we did a little simulation. Data were generated from the logistic map with 4 different values of the population growth rate, and those are illustrated by the colored bars. So we have 2 that would generate equilibrium, 1 that generates a 2 cycle, and 1 that generates chaos. For each of those different population growth rates, we generated 50 time points starting from random initial conditions, and then we fit the Gaussian process, which has now been extended to include growth rate as an additional input. So we fit all of the four different sets of time series all at the same time. And then we can use the fitted Gaussian process model to construct an interpolated bifurcation diagram. And so here's how that turns out. It's not perfect, but it's not too shabby either. And the cool thing about this is that it actually gets the period doubling in more or less the right place, even though we didn't have samples from that region. All right, so of course this again is just a simulation, and so it's a lot harder to apply these things in real life. So our next step is to move a little bit towards real life by applying this to a neat experimental time series in a terrific paper by Lutz Becks and colleagues. So in this experiment, they set up a 3-species food web growing in a chemostat. That's a little bioreactor where water and nutrients go in, water, bacteria, and waste go out. And the thing you can vary is the flow rate. And so by varying the flow rate, they showed that at high flow rates, we end up with a steady state for all three species. When the flow rate is reduced, we get chaotic dynamics. And at lower flow rates, we get population cycles. And that's illustrated on the bottom. And so if the only thing changing in this experiment is the flow rate, then these time series represent a one parameter family of dynamical systems. And so, as you know, that means that we ought to be able to just do the thing we did for the logistic map and reconstruct the bifurcation diagram for this system. And so we fit the Gaussian process to the whole experiment, treating the flow rate as one additional input. And then we varied the flow rate in the trained Gaussian process to visualize the bifurcation diagram. And this is how it turns out. The colored bars indicate the flow rate that were used in the experiments, and we get cycles, steady state, and chaos in the places where the experiment did. But interestingly, we also predict that there was a period doubling cascade for flow rates between 0.52 and 0.54, and I would love to do the experiment to test whether this is actually what would happen in the chemostat. But despite writing to him a bunch of times, Professor Bex never responds to my email. So who knows whether we'll ever get that done. Okay, anyway, so yeah, this time delay embedding stuff might be a black box, but you know, I think it's a really cool black box. Now, okay, having just talked about an experimental demonstration of chaos, I want to turn for the remainder of the talk to the age-old question of whether the dynamics of natural populations are chaotic. The idea that fluctuations in abundance, which are seen in most species, are the result of chaos rather than environmental forcing, was introduced to ecology by Bob May and Bill Schaefer and others in the 1970s. And since that time, there have been several attempts to quantify the prevalence of chaos in ecological time series. And the most recent of these is this paper in Ecology Letters that looked at 635 time series and found that just one was chaotic. At least that's what they concluded. And in fact, this idea is so firmly entrenched in the mind of ecologists that even the Oxford Bibliography entry on chaos in ecology concludes that chaos is rare. But really, how can this be? It kind of doesn't make sense. First, ecosystems are large, complex systems that are and such things are generally prone to instability. Second, nonlinear dynamics are ubiquitous in ecology. Everywhere you look, the dynamics seem to be nonlinear. And finally, and most glaringly, we all know that population dynamics are affected by the weather, and the weather is chaotic. And so it almost doesn't make sense that chaos can be rare, but and so a lot of people have spent a lot of time trying to explain why ecosystems are stable. But rather than try to explain stability, we decided to ask whether the apparent rarity of chaos is actually just a methodological artifact. The main reason is that virtually every meta-analysis that concluded that chaos was rare was based on fitting a 1D population model to the time series and then asking if the estimated parameters were in a region where the deterministic skeleton produced chaos. And so unfortunately, 1D models treat all interesting dynamics, such as species interactions, as noise. Now here's an example. The dots are some simulated data, and the line is a model fit to those. And because you're not ecologists, most of you, just so you know, a fit that looks this good would have the typical quantitative ecologist rolling on the floor in ecstasy. That's a pretty good fit by ecological standards. And the fitted model is stable. But the data were actually generated with a 2D deterministic model with chaotic dynamics. It's not noise, it's just another species that's been left out. And so I think it's fair to say that all the previous meta-analyses asked the question of whether one-dimensional chaos explains population fluctuations. And I'm happy to agree that the answer to that is no, but that doesn't really seem like the right question. Now, In the 50 years that have elapsed since chaos was introduced to ecology, a host of tools have been developed for classifying time series that don't rely on fitting a one-dimensional population model. However, these were mostly developed outside of ecology, so it isn't clear how well they'll perform on the short, noisy time series that we have to work with. So the first step is to evaluate how well these work, and then we can apply them to a relatively large set of ecological time series. So we evaluated 6 methods of classifying time series. They are shown here. That's 2 ways of estimating Lyapunov exponents: the recurrence quantification analysis, permutation entropy, the horizontal visibility graph, and the chaos decision tree. In order to test these, we started with 21 models that had either steady state, periodic, or chaotic dynamics. We applied a range of 4 different observation noise levels and 5 different time series lengths, and then simulated each of those combinations 100 times to determine the false negative and false positive identification rates. Then, in order to make sure that those rates that our classification success generalized well, we did it for a set of 11 new models. And then, because reviewers are never happy, we did it again for another set of new models that had both process and observation noise, but fixed time series length of 100. So overall, the classification rates look like this. No method gets it right every time. And there are 3 methods that were actually pretty terrible, with error, either false negative rates or false positive rates bigger than 50%. And so we threw those out. The other 3 methods were reasonably effective classifiers. Importantly, the performance, the classification accuracy was similar across the validation data sets. And we found, importantly, that observation noise generally increases the false negative rate, but not the false positive rate. And overall, for a short time series, the Jacobian method of Lyapunov exponent estimation was the best approach. So just in case, here's a brief background on estimating Lyapunov exponents. The Lyapunov exponent measures the exponential divergence of initially close points. So if we think about starting in the little yellow ball on the left and taking a collection of points there and following where they end up in the future, say the pink ball on the right, if the ball grows, then those trajectories are spreading out and the local dynamics are unstable. If the ball shrinks, the local dynamics are stable. If we measure this local expansion rate averaged over the attractor, and if the average over the attractor, which is the Lyapunov exponent, is greater than 0, that's taken as an indication that the dynamics are chaotic. Now, the Jacobian method for doing this constructs, well, Jacobians or matrices of partial derivatives from the delay embedding model at each time point, and computes the Lyapunov exponent from the dominant eigenvalue of the product of these over the length of the time series. So for ecological time series, we turn to the Global Population Dynamics database, which has more than 1,000 time series from around the world. But these are mostly quite short, on the order of fewer than 20 data points. And so we focused on time series where chaos might be detectable if it was there, which is, at least, and our criteria were that we had at least 30 time points, not much missing data, and a relatively high reliability score, which is, just a qualitative score associated with each time series in the database. So using those criteria, we ended up with 175 time series representing 138 different species. So here's how that turned out. Using the 3 reasonably reliable classification methods, we found that at least 1/3 of the time series were chaotic. Moreover, chaos was most likely to occur in plankton and insects, and chaos was least common in birds and mammals. And so I guess the first question that we should ask is, why is chaos so much more common in our analysis than in previous meta-analyses? So returning to our one-dimensional model hypothesis, so if we constrained our non-parametric approach to 1D, that is, we fix the embedding dimension to 1, or we fit models with only one input, the prevalence of chaos is reduced to 9%. If we go a step further and use the parametric models that were used in the previous meta-analyses, the prevalence of chaos is reduced still further to less than 6%, depending on which model you pick. Now, you might come to a different conclusion, but my take on this is that one-dimensional models stink at detecting chaos. Now, interestingly, chaos is most prevalent in short-lived species, and the Lyapunov exponents we estimated decrease with generation time. Now, that might be because long-lived species are better insulated from the environment, or it might be because we just don't have long enough time series for the long-lived species. Now, in addition, we found that the Lyapunov exponents, the positive Lyapunov exponents, scale with mass, not as a quarter power of mass that we might have predicted from the metabolic theory of ecology, but closer to 1/6. To see if this result generalizes, we estimated Lyapunov exponents for another 30 time series for zooplankton from lakes. Those are shown here in red. And then we added in some Lyapunov exponents that were previously published in the literature, mostly from lab studies, and those are shown here. Now, yes, this plot is not super clean, but I think that there's a case to be made that the Lyapunov exponents scale with mass across taxa, although the exponent is of 1/6 is an outstanding theoretical question. So to wrap up, I am going to say that here's my big mind-blowing conclusion here. Ecosystems are not one-dimensional. That's probably not a surprise to anybody. But the important point here is that ignoring this fact leads to models that mischaracterize dynamics and inflate noise. There's a big difference between starting out with a complex model and finding a reasonable 1D approximation using either a Poincaré section or timescale separation or something like that. Between doing that and just starting with a 1D model that we've just written down. And so if we want to get around the fact that we rarely have data on all the relevant state variables, it seems like delay embedding is a viable approach. Now, we've shown that although delay embedding traditionally is restricted to autonomous dynamics, we can extend our delay embedding models potentially to account for climate change, either by incorporating the mechanistic temperature dependence from the metabolic theory of ecology, or by sampling across distinct environmental regimes and reconstructing the bifurcation sequence. Oh, and finally, I showed that chaos is not rare in natural populations. Birds and mammals, although charismatic, are grossly overrepresented in our database. They were 60% of the time series that we had to work with, but they represent less than 1% of the species on Earth. Chaos was actually quite prevalent amongst invertebrates, and these are by far the most common things on the planet. So chaos is likely to be quite a lot more common than the 1/3 that we found in the global population dynamics database. Now, our next steps in this work include multi-species forecasting and multi-species management. I think the big issue here is actually the curse of dimensionality and developing scalable algorithms for multi-species management and multi-objective management. And so I'm actually a bit more excited to try our climate change and regime shift approaches on some field data. I'd be thrilled if you knew of any candidates. We've been looking for a while, and I haven't found any really good examples yet. And finally, I'd love to try an experimental evaluation of the bifurcation sequence that we inferred with the GP for the chemostat study. Of course, that means that I'd have to be able to get Lutzbeck's to call me back. Now, I didn't do all this work by myself, obviously. Lots of people contributed, some of whom you might recognize, some of whom know that they should smile when you go to take their picture, but not all of them. And I'd be remiss if I didn't thank the National Science Foundation, the Lenfest Oceans Program, and NOAA for their financial support. And with that, I'll take any questions.
Thank you very much, Steve, for your great talk. I thought that was fascinating. Can I start off by just asking, do you think if we did have more longitudinal data then on species like birds and mammals, that it would increase the chaos to be in line with insects if you kind of normalized it based on kind of generation timeline?
Yeah, it's hard to say. Your ability to detect chaos depends on the intrinsic time scale for the system, right? So the only way I could think of that we were able to get at this question was to take the time series that we had and shorten them and see how many we lost chaos in. And so if we do that and we cut everything down to just the minimum 30 time points, 40% of the ones that were chaotic now look not chaotic. So the time series length is a big factor. And so, yeah, I mean, it might be. On the other hand, there is some argument to be made that things that have a lot of age structure are much more insulated from environmental fluctuations. So I think that's only time will tell. We'll have to come back in another 20 or 30 years. So.
Yeah, fascinating though. Do we have any more questions from the audience? If anyone would like to unmute themselves. And okay, Matthew, Spencer.
Yeah, I have a couple of questions if there's time for them. But my first one is a really basic one about the Takan stuff, which I've never really spent any time on. So you've got your xt plus 1 is f of xt, where f is smooth and deterministic. So your underlying model is in discrete time and deterministic, but what if your real process is in continuous time and stochastic and also has measurement error? Is that any different from unobserved deterministic states?
Let's start with deterministic but continuous time, and then we'll talk about stochastic. So in deterministic in the continuous time system, to get to a discrete time thing, all we do is integrate, and we end up with the flow map for the system. And so Takens applies directly to that. As soon as we add some process noise, there are a couple of papers by Jaroslav Stark from the late 90s that show that with a stochastic driver, you have a skew product system, and you can do the time delay embedding provided that you have data on the driver. But there's also some other work by Holger Kantz that shows that you can actually achieve a time lag reconstruction in a stochastic system if you don't have that driver, but that's not generic. So that we lose genericity for stochastic differential equations. And some of the work that I've done with, say, birth-death processes shows that what we end up with is a reasonably good reconstruction of the conditional expectation. And so, yeah, a lot of the time it works pretty well, even though the theory isn't there to provide the same kind of guarantees that you have for deterministic systems. Was that a reasonable answer?
Okay. Does anyone else have any questions from the audience?
So can I ask, so this physiological time, or what do you call metabolic time step, is that the same as or related to what us old people used to call degree days? I mean, is that what you're talking about?
Well, so instead of just adding up temperature to get degree days, we're adding up the Arrhenius temperature dependence. So is it exactly the same? No. Is it conceptually the same? Yeah. And it turns out that it actually is relatively robust to what function of temperature you have there. Because one of the things that I had done for this project that I didn't talk about today is that a lot of ecologists or physiologists complain that the metabolic theory of ecology says that temperature response is just monotonic. But of course, for most things, you make it hot enough and they die, so the temperature response comes back down, right? And so people say, well, what if you did it with real thermal performance curves? And so there were a couple of species where I was able to get real thermal performance curves estimated. And the answer turns out to be relatively robust to whether we use the metabolic theory of ecology or a real thermal performance curve. The main difference is just the main thing is that we're getting the seasonality, that it's fast when it's hot, and we have a short time step for that. And then when it's slow because it's cold, we have longer time steps. So it almost doesn't matter as long as we do a reasonable job of capturing that.
So the conclusion is surely that you would say that in fact most species on the planet are chaotic. If you observe a third of them to be so, and you have that censoring effect of short data sets, you're inclined to believe that almost all, or not almost all, but certainly most species around are going to be exhibiting this. And I suppose I'm confused about why you think that is. Is it the inherent dynamics, or do you think they're driven by the chaotic weather, or does it make a difference to you what the mechanism is?
Well, there are a couple of things, right? Obviously, the weather driving things is a very plausible and simple explanation, right?
Sure.
Especially for why we would see chaos more likely in short-lived things
Sure.
than in longer-lived things. But there's actually a fair amount of, three simulation studies and theoretical studies that look at how the likelihood of chaos changes as you change the system size. Of course, they all have various assumptions that are made in order to make the theory doable. But chaos tends to be more likely the more number of interacting particles you have, or
Sure.
species you have, and that's my take on this. Is that I think that it's very likely that as soon as you have lots of things interacting, that complex dynamics are bound to come out.
So does that explain why you drew the cross outline through management? Because just as meteorologists aren't going to be predicting the weather in a serious way anytime soon because of the dynamics, we're not going to be able to control these species either, right?
Well, I think that, I wish I had a better reason than I just ran out of time. That's why I crossed out the management. But yeah, I think the management implication here is that certainly for things like fisheries, where we set policy based on steady state, that's crazy. And that a more reactive policy, like we would do with working around the weather, makes a lot more sense.
Or right? At least a risk.
Right. Yeah.
So I have more questions, but maybe Peter wants to ask something.
Okay, just a rather technical question, really. First, I must say, after working for several years with Gaussian processes, the performance of your models is really impressive, especially on the time series data and the bifurcation. Bifurcations are very, well known to be hard to predict with GPs, especially with the ringing that you see there. My question is, did you use any sparsity techniques to train the processes, or did you just go for straight, kind of map estimates for the hyperparameters? So like, did you have to account for the abundance of data, of simulated data that you had?
So all of these are trained on relatively short time series. I very very much spend most of my time in the ecological domain of not more than 100 points, and so I don't have the same sort of training issues with data set size that a lot of the GP work does. In terms of sparsification, what we do there is just the automatic relevance determination. So we set a prior on the length scale parameters that has a mode at 0. The mean for that prior is set such that the expected number of wiggles over the unit interval is 1. So I'm asserting ahead of time that the functions that I'm interested in don't have 5 turns. They don't do this. They just kind of either going up or down, or maybe they go up and down. But that more turns than that is not a reasonable expectation a priori. And so we build that into the prior, and that leads us to we really avoid overfitting, and we tend to do a pretty good job at identifying the relevant lags. So I did a couple of simulation studies looking at, whether it picks out the right lags, and that usually works pretty well.
And that's over how many dimensions?
So typically for time delay embedding in general, and sorry, I should have said this, the argument from the chaos literature in the 90s is that your ability to resolve the embedding dimension, or the max resolvable embedding dimension, scales roughly as the square root of the time series length. And so that's in a paper by Howell Tong. And so, so we say, okay, well, if I have 100 data points, the max embedding dimension is 10. And I'm just going to search, I'm going to have 10 inputs, and I'm going to allow the automatic relevance determination to pick out which ones are actually matter.
Okay, thank you. Thanks.
Sure.
I
have another question, kind of following up from what Scott said when you crossed out management and you said you ran out of time. I was wondering whether you could speak a little bit more about what you mean by a reactive kind of policy. So what's going on? Like, what's the implication of your research, and how would that actually influence policy? Because obviously this is really important work, and we've oversimplified for a really long time what the reality is. And this extra fidelity, what does that result in? How we can hopefully conserve more species in the future?
Well, so what we really need are state-dependent management policies, right? And that's kind of what I mean by reactive. If we know, so let me back up. So the standard sort of fisheries thing is to say, okay, we know what the biomass is now, right? And it's above or below our steady state baseline, and we set a harvest rate based on that. But if we know that there are lots of other species involved, right? Obviously, it doesn't matter just what the biomass for our focal critter is. It matters what the biomass is of the things that eat it, and what the biomass of its food is. And so we would need to have state-dependent policies, a policy that recognizes that these things interact. And I don't think we're going to get there by constructing ecosystem models, because I think that that's too big an ask. And so instead, what we've been trying to do is ask whether we can use our delay embedding models in the context of an optimal control thing to ask. If I knew what the, let's say, last 5 years of biomass were, could I construct a more robust harvest policy? And the answer there seems to be, we've done this for models of up to 5 interacting species. And the answer is that, on that scale, we can actually do a lot better than the sort of, the state of the art, which is just, 1 species at a time. I don't think that we're going to get to real optimal many species management with this. And the reason for that is that you actually don't see enough of the state space when you're trying to reconstruct the dynamics just from data, right? Like, you only see the parts of the attractor that you've seen so far, right? And it may well be that the optimal multi-species policy is well outside of that, such that we don't do a good job of interpolating with the GP. But at least we know we can do better than the status quo.
Yeah, great. Thank you very much. I think Matthew Spencer has another question. If you've raised your hand again, I'm not sure.
Yeah, if that's okay. So in the part on temperature, where you're regaining autonomous dynamics by switching from calendar time to physiological time, is there any connection between that and the idea of dynamic similarity in dimensional analysis?
That thought had never occurred to me, and I don't want to just kind of make up an answer. I don't know. That's a cool idea.
Okay, if you don't want to say anything more on that, Matthew, have we got any more questions from the audience?
I have a couple. I waited until the sirens went by.
No, they're still there. They're after you.
So when you looked at the Lake Geneva time series predictions, did you formalize the validation of those data, or is it just the look of them? Did you have a numerical characterization of the validation? When you said
so it's all based on leave 2 years out. So we ask how well we can predict the next year, given the previous time series, dropping 2 years worth of data on either side of the target point. We do that for the whole time series. I'm not sure what you mean beyond that. I mean, that's
so did you characterize the degree of agreement between the predictions and the observed data?
Yeah, so that the out of sample R squared is about 0.65 there. So it's not perfect by a long shot.
Okay. And so
but it's better than the fixed time step thing.
Right. Quantitatively better.
Yeah.
And I assume you've had students who've applied this to finance and stock pricing. So there must be such thing. So what's the analog of metabolic embedding for stock pricing, I'm guessing? Or I'm wondering.
I have no idea, but I should point out that this idea is extendable to any separable system, right? For all I know, as far as I know, nobody's actually done this in the delay embedding literature ever. But it applies to any separable system where you just change the if you have a driver and the dynamics are separable, you can change the time scale to eliminate that driver from the dynamics, and it doesn't matter that it's.
So what would that mean in finance, for instance?
Well, I don't know. What would that mean in finance? You'd have to ask a guy who knows something about finance. That's
So you haven't got a reaction from the, well, that prediction of the bifurcation is amazing. I'm not sure I believe you, actually. It's too good to be true. The guy at your course or your would-be correspondent clearly doesn't believe you either, maybe, right?
Well, that guy has never responded once. If any of you know him, send him a note and ask him to respond. But yeah, I don't know what the deal is there.
You could do the experiment yourself, maybe.
Yeah. So I did have a brief foray into trying to do these kinds of experiments myself, but all of my experimental experience is growing fish in buckets. And so when I started with rotifers, I was growing them in the same 5-gallon buckets that I'd grown my fish in. And when I talked to rotifer people, they thought I was totally out of my mind because they grow them in little tiny containers like this.
Right.
I responded by saying, well, at least I don't have any, a demographic stochasticity.
Thanks.
Some of the participants also wanted to know if they wanted to reference your work, how could they, what should they reference? What's the best way to, any papers that you have on this current work?
All of these I think are up on my webpage. And if they're not, anybody can feel free to shoot me a note and I'll send them reprints or of whatever.
Okay. What was your website? Sorry, just for
It's
so I can check it out further.
Yeah, that's a good question. What my website is? It's yeah. Is there a way for me to write this? I can put it in the chat.
You can put it in the chat, or alternatively, if you haven't got it to hand, I can also send an email to all the people that attended.
All right. All right. So that's the website.
Okay, great. Do we have any final questions?
I think.
Okay. Well, I'm gonna check it right now and make sure that you haven't just given us the wrong number. Maybe you're just trying to
That's right.
off, you know?
Like, look over there. Yeah. So
you're not allowed to copy and paste anymore. Sorry. So does anyone else have any final questions before we wrap up?
Yeah, I have another one if that's okay.
Okay.
So about what you can actually do with management in big systems. And part of the choice between your approach and the kind of Levin's qualitative approach is going to be down to whether you're a Marxist or a capitalist, I guess. But I'm wondering if there's any way to bring those two things together. So can you go from what you get out of your approach can you bring that together with qualitative knowledge about the effects of parameter changes to make better qualitative predictions than you could do just with the kind of Levin's approach on its own?
yeah, that is a really good question. So one of the things that I don't actually have a direct answer to that. I think that's just a really important avenue for thinking about more. One of the things that we've been thinking about a lot is that this time delay embedding stuff is on the one hand great because you don't need to know how the system works in order to use it. On the other hand, it's not the same thing as saying that we don't know anything about how the system works. And figuring out ways to build the things that we know into these models is, I think, the most important thing that we can do over the next half a decade or so. And this metabolic theory stuff is my first step in that direction. In other things that we've been working towards building recurrent neural networks. So you can show that the Takens argument leads you directly to a recurrent neural network. And if you were able to say something about the functions involved, you can build that directly into the neural network structure. I didn't have any good results to show from that work yet, other than a bunch of stuff on pencil and paper. But I agree. I think that's a very important direction to take this stuff.
Okay. Did you reply, Matthew? Sorry, your mic's muted.
No, I think that I don't know how I would do this if I was trying to do it, but I think what you're talking about sounds really sensible.
Okay, I think we have time for one more question, if we have it. If anyone wants to unmute. Sorry, my phone is doing something weird. Any final comments or questions? No? Okay, well, thank you very much, Steve, for your talk. I know we had quite a long awaited kind of suspense building up to this with the protests and everything. So, but it was definitely worth the wait, and I learned a lot of things. So I'm really grateful that you've come here today to talk to us.
Thank you for having me. It was a pleasure chatting with you guys. And yeah, I really appreciate the invitation. So.
And finally, yeah, so your website does work as well. So if people want to reference your work or check out more of your work and see what your lab is up to, they can do so. And we'll be recording or uploading the talk onto our website as well. So we'll send you the link to that.
So all of my missteps can be preserved in perpetuity.
And mine.
Excellent.
Cool. Perfect. Well, thank you very much, everyone, and have
Thanks a
lovely
lot.
evening. Thanks. Bye.
Bye. Have a good night.